our esteemed professor and now friend, Pat Ewanchuk, is going to be talking about salt marsh loss and the work that he and Mike have been doing for the past 5 years. And Pat comes to us by way of Brown, and where he was a student under Mark Burtness and is now a professor himself at Providence College and the chair of his department, as well as an adjunct professor as well at Brown. So we are really happy that he's here and that he has committed so much time over the last 5 years to look at this issue and help us develop, we hope, some strategies and some ways of thinking about salt marshes that will educate us and bring us some sense of our future with the salt marshes. So Pat, thank you so much for being here today and for participating over these past years. It's been quite something to have the chance to go out and see what's going on out there with all the different sites that he's been working on. So let's welcome Pat.
Thank you all. Yeah, 5 years interrupted by COVID,
Yeah.
so it doesn't seem like that long, but it's 5 years. I put this up to start just to remind me that I want to thank the Westport River Watershed Association right off the top. It's been amazing working with you all, especially with Mike and Roberta and Deborah. It's been great. Also, can't forget to thank my colleague who did the first half of all this with me, Catherine Matassa, who's a professor at UConn. So she'll be thanked again at the end, but I want to make sure I get those in right off the top. What I'm going to try to do today is catch all of you up on the salt marsh, a little bit of history of salt marshes and where we are in New England with it, but more specifically Westport. Talk about phase 1, which was the early work and still continuing across the whole watershed, or not watershed, the river parts. And talk about phase 2, which is more the work that I've been doing with Mike recently. And then talk a little bit about what's next. Where do we go from here? What can we do? And what are potential solutions to what we found? So, salt marshes are this amazing estuary and habitat. And the way I think about salt marshes, it's all about the plants. If you don't have the plants, you don't have a salt marsh. You have a mud flat. And so it's really, really important to understand how those plants grow and how they accrete this wonderful thing, if you've ever been out on the peat base. The thing that when I take 6th graders out and I jump on the marsh and they all feel those vibrations, they get it. They understand that this is what we term a biological or biogenic community, that without the plants, when you jump in mud, what do? You sink. So those plants and their roots and all of that sediment that gets binded together really makes a difference. And that'll be pretty important because a biogenic community is something you all probably want to go vacation in. It's like a coral reef. Without the corals, you have a sand flat. And no one goes to vacation on a sand flat. They go because of the corals. And with those corals, you get the fish, you get everything else. And so the salt marsh, think of it, same as your coral reef. It's the plants or the corals. With those plants, they make that community. You get all the other things that when you go to a salt marsh, you see the birds there, you see the crabs, snails, all the other cool things that are there, but it's plants first, okay? So we're going to talk a lot about plants in this thing because that's my passion. All right, so plants, these plants that make up the salt marsh, they're there, and as they grow, part of their productivity or their biomass gets deposited there, and it gets tangled up in their roots, added with sediments that's in the water column, and you build up this incredible marsh peat, okay? So that's really important to know. Those sediments get bound up and tied there. And you'll see, so this is what my undergrads hate when I do. I'm going to tell you 2 things you have to remember for later in the talk. And they all say, oh, Dr. E, why do you have to do that? I can't remember anything you say until I study it. I'm like, for 45 minutes, I think you can remember this, okay? So this is what 2 things I want you to remember. It's the plants that build the peat, right, by those little sediments and all that stuff getting deposited there. The only way that stays there is if it's in the right set of water conditions. When the hydrology or the water moves too fast, all that stuff gets taken away. Okay, so that's number 1. Number 2 are these little black dots on the bottom edge of this thing which indicates the rib muscle dukensia. The rib muscle in New England salt marshes is a vital part of our salt marshes. My mentor, Mark Ness, did a lot of work on sort of positive effects of species on each other. And one of the things in the salt marsh he worked on is how Spartina alterniflora, the main cord grass down here in the low edge of the marsh, has a positive relationship with that rib muscle. Where you have the rib muscle, you have really good Spartina growth, and you have a New England salt marsh. If the rib muscle is absent, you still might get Spartina. But those marshes aren't typical, healthy New England salt marshes. So rib muscle, really important to remember. And two, that it's a biogenic community for those sediments. Remember that will be good, I think. I'll probably forget, but you guys will remember. Okay, so what does the salt marsh do for us? Well, obviously it provides habitat for all those coastal organisms, both invertebrate, vertebrate, birds, fish, all that kind of stuff. Really important. That's why I love going out there to see stuff. They also protect coastal communities from extreme storm events. They're a buffer, so when those waves come ashore, they're dampened by the presence of that biological community. So if you sit right on the front edge of a marsh, you get hit by a wave, you back up. Even if that wave rolls across the marsh, it's attenuated, it's slowed down. And that's a really positive thing for our coastlines. Okay. They're also really important for both commercial and recreational fish and shellfish industry. Okay. Plants the bottom of every food chain. So as you cascade up to what we want to eat, it's important. Okay, so that's another part that's really important. And then finally, they're a storage of carbon. This has become sort of more of a focus as carbon and climate change has become more important. And our realization of where are the carbon sinks? What is sucking down CO2 out of the atmosphere? And as you'll talk about in a second, salt marsh plants are a big sink for carbon. Okay, they're really fast at taking carbon out of the atmosphere. Okay, and they store a lot of it. So the plant that does the most of that carbon storage is actually Spartina alterniflora, cordgrass, the dominant grass you see on the edge of the marshes. Okay, it does the majority of it because it's the biggest, fastest grower. You get about 3,300 grams of biomass per meter squared. All that doesn't mean, just means it's productive. There's a lot of grass. You don't care about the numbers. I don't care about the numbers. But this is where it's really important. Marshes aren't all equal. Okay? And the rate at which Spartina grows is highly determined by the water flow. The delivery of nutrients to it, how much is taken away, because every plant likes to sort of build that peat up, store some of the stuff there, and they use some of those nutrients again. And when it's all transported out by high flow, you get a different rate. So it's basically what we call hydrologically dependent. Okay, so again, water flow is hydrology. So I put my little lightning bolts there because that's one of the two things, right? Did you forget? No? You got it? All right. So the lightning bolts are to remind you, one of the two things. Okay, so in New England, we have a history of impacting water flow in salt marshes. Right? We've been doing this for a long time. This is nothing new, unfortunately. Historical agriculture across New England has ditched and dredged marshes to grow mid-marsh salt marsh hay or Spartina patents. Okay, so we spent a lot of time ditching and dredging to increase it and then cutting that. All of that has affected the water flow around, in and around marshes. We've also done mosquito ditches. Okay, so for a long time we thought that we had to drain the marshes to get rid of mosquitoes. Until we figured out with an entomologist that mosquitoes don't nest in salt water or don't lay eggs in salt water, so that was a waste of our time. But nonetheless, it put a lot of people to work for a while, right? We did a lot of work. Trust me, digging all those ditches was a lot of work. I didn't do it, but I know how heavy that stuff is. Tidal restrictions, roads, all those kind of things have also changed. Coastal development, all have changed hydrology in coastal marshes. Okay, so we've been doing it for a long time. We have historical pictures to show us out there doing it, right? And it doesn't take much, but you can fly around if you have a helicopter. Anyone got a helicopter? But if you fly around New England, you can see it. or take my drone up and you can see it. These straight lines are not natural. The history of our changing coastal hydrology is evident, okay? And so those changes to hydrology have not only changed nutrient flow through a salt marsh, but they've also changed the water, or freshwater throat. Ugh. It's tough, I've lost a tooth, so now fresh water flow through a marsh, right? So more is impounded, more is access to larger areas. It's just changed water flow. And then all of this is now basically compounded by the effect of sea level rise. As sea level rises, more water can get into the marsh, and with all of those nice ditches and channels, can more infiltrate different areas that faster and for longer than it ever had before. So where are we now? So this is one of my, unfortunately, favorite pictures from New Jersey, relax. But this is what we've done to salt marshes. Because coastal views and coastal property is great. This clearly was a marsh, and there's still marsh. There's still marsh there. But you also notice on this side, there's this big barrier. And it looks kind of natural, but that's a man-made barrier to protect that marsh on that side. So not only have we lost marsh, but we've changed hydrology again. Okay? So worldwide, this is not a New England problem. We're losing marshes, marsh habitat worldwide for lots of different problems, lots of different reasons. It might be changes in hydrology, power issues over in the Netherlands where they're gating and using hydropower, introduced species, there's lots. In New England, our major threats are eutrophication, salt marshes, shoreline development and changing of hydrology, predator release, we call it, but basically a result of overfishing down on the Cape. There's not as many coastal fish coming in, they're not eating the crabs, and Sarsima, this little purple shore crab, is eating marsh roots now. You may have heard that story, but we'll talk about that too. And then climate change and sea level rise. So there's lots of different threats. It's estimated today, though, that from the time the Pilgrims landed in New England, we've lost about 80% of our coastal marshes. So the marshes you see out there are our remaining 20, and they're under threat. So if you put that in your mind, that we're dealing with the last 20, and if the marshes are this valuable and this important to us, that last 20 should be high, high priority. So what is the value? So some smarter people than me, because I just deal with plants. So economics, or economists, that's the word I'm looking for, have looked at natural resources and all of these functions that salt marshes play a part in, and tried to estimate the value in something we can all relate to, which is the dollar, right? I think of it as it's invaluable, but marshes can put a number on it. For instance, we can say the protective value due to storage surge in order to keep people's property safe runs around $7,000 to $8,000 a year per acre. Okay, and we can do the same thing for carbon. How much carbon is taken up by a marsh per acre, and estimate that in terms of carbon credits and other things like that, and come up with a number. And we can come up with that for all of those things, and it's estimated somewhere around $10,000 to $24,000 a year per acre is sort of the ecosystem service value of just having a marsh there. Okay, so with that number and the remaining 20%, we want to protect it. It seems like anything that impacts those marshes, we should invest in restoring it or keeping them healthy. Okay, so I looked at some of the values to fix a problem, right? So we have a marsh degrading somewhere. What would it cost to restore it? And so there are estimates. And to restore an acre of salt marsh is somewhere, the lowest I could find was $16,000 per acre. Okay? Which that includes planning, construction, and monitoring to make sure that your restoration did something. Those numbers, I saw numbers up to 1.5 million, not per acre, but the project was like 1.5, $2.2 million, and it took the acreage out of there. It's well into the $50,000 to $60,000 an acre. Depends on what type of restoration you need to do. So, obviously, not all restoration is the same. And really important in restoration work that you need to be able to fix the problem. Okay, and this is really important. Think of it, you take your car in to someone, and his only fix for your car is to change your tires. So you take your car in because it's not starting, and he goes, all right, I'll fix your tires. And then you leave. What value have you gotten? None. So there isn't just one fix to fixing a car. Why would we expect one fix to fix a salt marsh? Right? We shouldn't. Yet, there are things, and I'll talk about at the end, that unless you know the problem with the marsh, right, you can't fix it. Okay. So let's think about what's going on here in Westport. So the Westport River, we've seen declines. It's well documented from aerial photos and stuff. Costa's paper years ago showed these declines. And it looks like there's an accelerated rate in the last 20 years. And this is what drew me into working in the Westport. What is going on here? Okay? And it also showed that the most rapid decline was in the lower reaches of both rivers, and the West Branch for sure. And you guys probably have all been out there and seen it, as being good people from Westport, right? So, but there are other areas that aren't so bad, right, where the marsh looks pretty good. And so, what is driving this loss in the Westport River, and specifically what's driving it in different areas? Why aren't they all the same? So years ago with Catherine, we sort of divided the river up, or the rivers up into, obviously, West and East Branch. We didn't do that part, but we did upper and lower. We picked a lot of sites, 14 sites, 7 on each branch, and really started to get into this problem. And we had 2 leading hypotheses just from our surveys of the marsh, which, when I say surveys initially, we just drove around. I think Mike and Roberta were nice enough just to take us around, drop us at marshes here and there, and figure out, what do you think of this one? And I'm like, oh, this is a beautiful marsh. And then I go to another one, and I'm like, yeah, what's going on? So we thought maybe it's nutrient loading, and I'll explain why we might have thought of that. Okay, that basically nutrient loading around that time had been shown that if the plants are nutrient loaded, they don't invest in their roots because they have enough roots to get, because there's a ton of nutrients there. And if little roots, then all of a sudden any kind of change and they collapse and you lose marsh. Okay, and so there was this destabilizing effect of nutrients. So we were like, okay, we can look into that. And maybe because this is a big watershed with lots of different currents and things, maybe water flow or hydrology matters. So our approach was to basically start off in phase 1. We basically did a bunch of surveys, and I'll explain what that means. But we went around to all those marshes and looked at both the abiotic and the biotic parts of a marsh. So basically the biology, and then things like flow and sediment load and those kind of things that are non-biological but important for biology. And then we did some field experiments, a massive nutrient addition experiment to look at the nutrient question. And then what we learned from that, we stepped into phase 2, which we'll get to, which was quantifying the hydrological differences across the marshes and across both branches of the rivers, which is a massive undertaking, as you'll see, and then tried to correlate those changes in hydrology to marsh health. There's some indication of healthy marsh and not so healthy marsh, we'll put it that way. Okay, so thinking about this nutrient idea, previous work of mine and Mark Burness and Brown, my PhD advisor, showed that, and I'm summarizing like Mark's whole career in one really bad slide, so I apologize if anyone knows Mark. But basically we showed that if you increase nutrients, the plants do better. And it's like, duh. But no, there's a little bit more detail than that. And that those growth changes actually change the competitive dynamics in a salt marsh. So plant that was a bad competitor under different nutrient load becomes really good competitor. And it changes what the marsh looks like. So we had done this work, and then there's some, obviously other people working in it, and Linda Deegan up in Plum Island did this fascinating 9-year study where she dumped a lot of fertilizer, this is one of her boatloads going out, onto a marsh. So they exceedingly loaded a marsh with nitrogen and said, what effect does lots and lots of nitrogen do? And what they found is it actually led to marsh degradation, which was kind of interesting. And this is that root idea, that if you pound this with excess nitrogen, the plants don't need the root system as much anymore, because there's no need to look for nitrogen, it's everywhere. Okay, so they don't invest in their roots, they grow, right, because they have all the things they need, sunlight and nutrients. And then eventually what happens is the marsh just collapses because the roots aren't binding that peat together. Okay, and they have evidence up in Plum Island. These are the non-loaded marshes, and you see the marsh bank. And then the loaded ones, you can see it crumbling and falling in, basically what's called caving. It's like what icebergs do when they think that's called caving, or calving, I don't want to say that way too. Anyway, so this is what she found. And so when we first stepped out off the boat in Westport, this is what we found, is these big chunks falling in, and you guys have probably seen this in the lower west branch. Okay. But something different's going on here. It didn't take us long to realize something different's going on. Looks the same superficially, something different going on. What is that? Well, a couple things you can see is, in this picture where I'm standing here, it starts to round over. So the edges were slumping. They're being undercut. So you can actually, if you're brave enough, you can actually snorkel a little bit underneath here, which is kind of scary, because that edge is sort of teetering. And then every once in a while, it gives way and cracks off, and you get these big chunks that are sitting there. Okay? So we have the calved areas and these sloped areas that are undercut. And I'll show you some better pictures of the undercutting in a bit. Other thing we noticed right off this, I mean, it didn't take as much. We jumped off the boat, and you basically hit really hard sediment, like boom. And I'm like, wow, that's nice. I don't sink into the marsh. I didn't need my hip waders today. It's so hard there, and I'll point this out again later, that this snail, Littorina littorea, it's our New England rocky intertidal common periwinkle. It's not supposed to be in a salt marsh. Well, it doesn't know that, but it's hard enough because it doesn't like mud. So a salt marsh is typically muddy. That one would be trapped in the mud and would not want to be there. But it can graze around on the surface of these marshes because it's so hard. There's actually, I've got a better picture of these, are juvenile barnacles. You might have been scraping off the hard bottom of your boat. Something that also that lives on rock, not marsh. They're hard. Okay, so we saw these differences. So, quick reminder here that a salt marsh is a product of the species that are there, the biology part, and its physical environment. Which means, although marshes are marshes, they all have spartina, whatever, they look the same. They're really different. Okay? So that's our challenge, is figuring out what's causing these differences. So I'm going to show you, even here, you probably haven't driven the coastline lately. These are all from roughly the same week a couple of years ago. And I'm going to highlight the spot in red over here of where we are in your watershed or in the river system to what the marsh looked like. So this is way up West Branch, healthy marsh, nice tall spartine alterniflora. We go a little bit south, still pretty good marsh. Looks not quite as tall, but pretty good. We drop down into the lower branch, and you start to see this wavy, caving, weird overhang marsh. And note, this is like a putting green comparatively, right? I mean, you can chip off that, no problem. You try chipping in the marsh upriver, you'd be swinging like you're farming or something. So, and that's also where we see these big chunks falling off as we go down the river. We turn the corner, lower East Branch, so hard again that we see, you can probably see a couple of these guys, I don't know, probably not in the back, but those snails appeared again all over the place. Grazing on the top of the marsh. That folding thing, and then I didn't bother going back up the East Branch, but if you go back up the East Branch towards Hicks Bridge, it starts to get nice again. Okay. In my mind, when I was seeing this, I'm like, but Roberta just told me highest nutrient load in the river is where? It's way up here.
Yeah.
So we're nutrients? That doesn't make sense when we're seeing the degraded marshes, lower river, where the nutrient load is low, and then nice marsh up where the nutrients are high. So that's just observation. We need to do some testing. Okay, so what is driving it? So we did these surveys, and I'll explain the details in a second, but we looked at plant growth. We looked at benthic invertebrates, which just means the invertebrates on the marsh. We looked at water flow, crudely at that point in time, sediment compaction. Then we did this field experiment. So our survey techniques were based on this one sort of overarching hypothesis that degraded marshes are the ones in the lower branch that didn't look as good. I hate using the term degraded. I probably shouldn't take it on there. Not so good marshes, we'll put it that way. Right? Especially the ones in the West Branch, out in front of the marinas and stuff, should be in areas of greater water flow, so we should be able to see that, maybe the hydrology. The sediment we needed to show was actually higher, or harder, right? So he's just saying that it's hard and I jumped on it. I need data, because that's the way I work. The Spartina was less dense and shorter, so we needed to measure that. The abundance of that snail, the common periwinkle, little rhino, would be higher in the degraded marshes. And then point number 1, I think, or 2 that I told you off the top, Ewenchuk, the rib mussel, you should see very few of them in the degraded marshes and more of them in the healthy marshes. Okay? And that's just to show you up here, this is what Ewenchuk looks like. Those of you who've walked around that muddy thing probably hear them cracking if you've walked out into a marsh, unfortunately, but really important for New England salt marshes. Okay. So our first attempt at looking at water flow was really crude. I said we used dental plaster, made these little hockey pucks because I'm Canadian, because I can relate to that. We take the hockey puck looking dental plaster things. My dentist loves the fact that I steal dental plaster from them. And the reason we use dental plaster is because it dissolves really slowly. Okay? So we make these little hockey pucks, we cut them, we weigh them, we put them out in the marsh, and then you can deploy them everywhere for a certain amount of time, and the rate at which they dissolve away, you can sort of basically back calculate that to higher water flow, more dissolved. Less dissolved, less water flow, right? Really crude, okay? So we did that, and it looks like, wow, well, in our downriver areas, both in East and West Branch, there's higher water flow or water movement, okay? Just because there was more plaster dissolved. That's all this is telling us, is at least we're in the right ballpark there with water flow. That's the difference between these sites. Our biological surveys are a little more detailed. We went to each of the 14 sites, did lots of replicate quadrats, 6 in each one, where you basically count every stem in that quarter-meter square. You measure the height of every stem. You look for every little snail or other invertebrate in that quadrant and get all that information. And we also tested sediment compaction in those quadrats. I'll show you how we did that. Okay, so we did lots of replicates. Roberta that summer was going nuts, driving us around, dropping us off, picking us up, dropping us off, all over the place. Okay, so Spartina ultriflora. These are the yellow is sort of is the upper. Sorry, I should have had this on here. Maybe I do. Nope. Is the upper river. The purple is downriver. So in the degraded marshes, the height of Spartina is smaller, taller, better growth in the upper branches. And you can see that here. Difference Sediment compaction, we used a penetrometer, which is basically a spring scale. So if you've ever like held up your prize fish and it basically pulls on it, well, we use that same type of instrument. We turn it around, and if you push on that spring, it reads or records here. So you're pushing this little 1 square centimeter disc into the marsh, and it measures compaction, how tight it is. There's a version of this that I actually bought that greenskeepers use to measure how hard the green is, but that one didn't work in the marsh. It was expensive and just didn't work. But this works pretty well. And downriver, they're harder. But the yellow one, the upriver, is not quite, it was weird. We were like trying to figure out. And it turns out those mussels upriver impacted our ability to measure hardness because we kept hitting a mussel. And we're like, so that yellow bar should be, you know, if we were actually being able to squeeze in between in the mud, and I had my undergrads doing it right, not just using the data, we probably would have seen a better result there. But there was a consistent pattern, so I'm going to have to say, believe me on this one. But I can take anyone out and have them sink in the marsh upriver if they want to come. Okay. Our biological surveys are really interesting. So we, again, went to those same things. We counted all those things. So we did the rib mussel, littorine. We actually looked for Sarsaima, this crab that's eating up the cape, see if it was there. Okay, so Gukensia, that mussel, right? Upper river, in the nice marshes, lots of it. Lower branches, none of it, or virtually none. You find one every once in a while. Okay, so that tells us two things. Those marshes aren't typical New England salt marshes and doing really well. And two, it's really hard for them to get into that sediment. They like to live in the mud, in that soft mud. And if they can't get in, they're not going to grow there. The exact opposite pattern for that rocky intertidal periwinkle. Lots of them downriver on that hard surface. Virtually none, well actually none, where they're not supposed to be, which is nice, upriver. And just to let you all know, we didn't find any of those crabs, so that's good. No matter how the marshes do them, they haven't invaded here yet, which is great. Okay. The other thing that shocked me, because I come from a plant background, is there's this green or brown algae called Fucus vesiculosus. You might know it as the common rockweed. It's all over the lower branch salt marshes. Okay. It's called, common name, rockweed. And it's growing and settling. These are all new recruits, these little yellow things. That is brand new rockweed settling on a marsh. Not supposed to happen. Okay. So, overall our impression is something's wrong on the lower branch. What's going on here? We did this big experiment, this nutrient addition experiment, across all 14 sites, where we basically divided them up, had replicate plots. You guys really know the mechanics of a big thing, but it's a lot of work. But I will show you this. How do we add nutrients? Some of you might remember if you saw the talk that Catherine and I gave. But we drilled these little test tubes with holes. We pack them with fertilizer. Some of them we pack with just aquarium gravel. Those are our controls. Okay? And then we insert these little plugs, essentially like fertilizer spikes that you might buy at a grocery store. But the reason they're packed this way is so we can actually measure the amount of nitrogen we're putting in. And because we pack them in these little things, that they're really slow to release. So it doesn't all just dissolve away on one time cycle, because that wouldn't be important. So we put them in arrays like this, where they're fertilizer spikes right into the marsh, and they slowly dissolve over the growing season. And you can do them fertilizer, not fertilizer, and have all those nice controls and do that kind of thing. It also means you got to drill lots of holes in little test tubes, which is always fun. Okay. Our control plots, so these are just plots where we monitored at all 14 sites, and we saw that total above ground biomass, or the plant growth, is higher in the upper reaches, lower in the lower region. That line that we put there is our artificial upper and lower. So it's not perfect in every case, but the trend is there. The upper branches have nice, tall spartina. Okay. our experimental data, I'm going to click here, in all cases, all you have to see is that black line is above the other line. That's fertilizer additions. Okay, so wherever we added nitrogen, the plants did better. So what does that tell you? That tells us that they're not nitrogen loaded. In other words, these plants can take more nitrogen if you can provide it. They can grow more when you give them more nitrogen. That does not say dump all your nitrogen in the marsh. That just means they're not nitrogen loaded like Deborah or like Linda Deegan showed up at Plum Island, right? She gave them so much nitrogen they couldn't take any more, and so you don't see any kind of a growth response. Here, if you give them more nitrogen, you see a growth response. Okay, so this is without nitrogen addition, with nitrogen addition. Okay,
And what about the roots?
the roots grew more as well. Yeah. So, and it even happened in the lower branches, but not to the same degree. So if you see this little, we like to call it our chia pet, that little bright green spot in the middle there, that's where we added fertilizer, right? So those plants responded positively, but not quite in the same way they did in the upper reaches of the river, okay? So yes, still nitrogen limited, right? But not in the same way. Okay, so our part one conclusion of why we got onto this bigger thing about hydrology is that downriver marsh loss is not nutrient driven, okay? Because they're not loaded, they're not, you can add nitrogen to do better, and the general trend across the river is that the high loading of nitrogen is upriver where the marshes are healthiest, okay? Then, you can see this ledge here, underneath here, that sort of scary part where you don't want to go snorkeling. Okay? That's that overhang. You can see it getting eroded away. You see the front edge of these marshes nice and hard, which means they're hard because the sediments aren't being allowed to build up there and accrete like marshes normally do. They're being taken away by the hydrology. So not only is all that, whatever production is here, it's supposed to come back and keep the marshes nice and healthy and building up, being eroded away, but even this top surface gets sort of scoured away. It's like having like a hose run across your driveway or your lawn for a while. It'll carve away, right? It's just going to continue to carve away that channel and take those sediments with it, okay? So all of this pointed to the likely thing that hydrology is some kind of a culprit and is probably leading to that sediment compaction, getting it harder and harder, or harder, and it also is indicated by that biological community where we're seeing things like the rockweed and those intertidal snails in this area. So what we thought is that why is it so hard? It's just the sediment dynamics. But we need to show this flow thing a little bit more detail, right? Because we just use my hockey pucks and everyone doesn't really believe them. Okay, so if flow is leading to sediment dynamics or changes in sediment dynamics, we should be able to detail that flow across these marshes and correlate it to marsh health. Okay, so that's where we get to phase two, right? Understanding the hydrology. How am I doing there, Roberta? Good? All right. Inundation time, because that's how long those marshes are underwater. These are terrestrial plants that are living on the edge, right? So they don't want to be underwater too long, but underwater for a while is okay. Is that increasing with sea level rise maybe? And how do you measure that? And then water velocity. So we picked out some of our sites. We picked 4 in the West Branch to sort of span that good healthy marsh to poor marsh. Picked a lot more in the East Branch because Mike had some other agenda. He had some reasons, we'll put it that way. And he was doing a lot of the work, so I said fine. So we picked a lot, and some really new ones, which were cool. We're going back up in the Let here and looking at a couple other sites. Okay. So literally Mike put to work at this point. Because in my mind, flow is, well, it's an engineering problem, for one. It's an expensive problem, and I really, many of the tools that I have didn't have anything that could do flow at the size and capacity that I was envisioning, which is both rivers, all sites. Let's do it. Plus, you don't do it just on one tide. You want to do it on multiple tides, and you can't do it one tide here, and then 2 weeks later, another tide, and then 3 weeks, because you guys know weather changes, tides change, all that, you need to do all, and almost all at the same time. That poses a big problem for low-budget ecologists like myself. So Mike and his crew of himself, with support from Westport, developed, first off, tested these tide gauges, okay, and flow gauges, which is incredible. So just surfing the internet, I don't know what he did at night or how long he spent, but he figured this out, okay? And then we were able to convert sort of pressure, water level to inundation time and get current measures and all that kind of stuff. So first off, you got to build these things, which he did in his garage, had to move the cars, figure out how to auger them in, deploy them at different sites, visit them all the time to calibrate them and be able to download them. He'll probably give me trouble because I'm going over this like this is easy. This is really difficult. And then, once they had those going, they conducted surveys across the marsh, so you could look at current flow and tidal height to the profile of the marsh, which is going to become really important. So what height each of the plants was at. Okay. I helped, again, conducting some biomass surveys, and I'll explain this again in a second. We calibrated all of those stations to NAVD 88, which is sort of the standard of the zero line of what low tide is. So when we get a tide chart and it says it's at zero tide, we had to make sure that our tidal gauges were calibrated to that. Not an easy task. You need professionals like mine. Okay, and then those all have to get removed. So lots of work. So just to show you just a couple more things. This is a stilling tube that went over, so when Mike was actually getting his readings, the waves weren't trashing his readings, so that had to get put on every time, so that's Mike lifting it over the top while he's on the boat, hopefully not falling in, which will be a prelude to something later. But getting all those data, okay? Doing the surveys means you gotta get out there, run a transect across the marsh, look at elevations that are now set to your zero tide data, and actually figuring out how many meters in across the marsh have I raised 1/4 of an inch, or have I gone up an inch, or am I down? What is that new tidal elevation? So this is Mike in the water. He got down in the water, right, to get some of these measures. That's nothing, because some of us actually get in the water, right? So I'd like to say I dove in after some piece of fucus or a snail or something, but actually I was standing on the front of the boat, I don't know, just looking around, and I took a step backwards and went right off the front of the boat in the water. Thankfully, it was a warm day, and Mike saved me. But, this also, just a note, if anyone's a PC alum or a Brown alum, this is a gifted shirt. I wear it in the field only. I didn't go to venues.
If I could just add here, I wanted to make a point that Patrick blends very well, okay? So he's been out on the river and he's seen all of these Westport guys standing up in their boats with the long connection to the throttle and just driving along. So he thought he could do that on the bow of the boat.
He actually rammed it in reverse just so. All right. So at the end of the growing season, I went out, and that's when those pictures were taken, and we did this end of the season biomass, which means we cut down the standing crop. Okay, so you put your quadrat down, you cut everything like a little lawnmower, you take all those stuff back to the lab and quantify it all. Okay, so these were done at specific distances from the edge of each marsh, which thankfully we now have tidal heights for each of those distances. So we knew tidal height of the plot. We could then say, at this plot, what's the submersion time? How long is that plot area underwater over a tidal cycle? And what is the relative current speed at that? So lots of really cool data from these flow and tide gauges.
So this is what my lab looks like at PC in September, October, November, December sometimes. It's full of plants. So every bag is a biomass sample from a quadrat. I spread them out all over the place, start to sort through them one at a time. So they dry, I sort them, count them, measure them by stem, then it all gets cut up, and then weighed, and we finally get some data. And I think Deborah and Mike now have an appreciation after working with me for 5 years. It just takes some time. At first they're like, oh, just take your sample. Then they'd call me and like, you got data yet? Jesus. It takes some time. So we get through it. My colleagues hate this time of the year because the marshes stink when they're drying in my lab. So there's this nice smell of dead, rotting grass coming down the hallway. Whatever. So we finally can get to a graph after all that. And so this is biomass of Spartina alterniflora at the edge quadrats, so not all the data, I'm not going to show you everything, by what we came up with the hydrological index, which is inundation time, so how long it's underwater, so the percent of the tide cycle it's underwater, multiplied by water velocity. So it gives you, while it's underwater, what flow is it seeing, and how does the biomass do. And this is at, I think, 12 sites, and you can see that there's this negative slope. But what's really cool is for the 50% of the variation in biomass is explained by inundation time and water velocity. That's got nothing to do with nutrients or sediment type or anything. Those two variables explain 50%. And it gets even better if you take inundation time out and just look at water velocity. Okay? So yes, water velocity correlates to compaction and all those kind of things, but that's the driver, is water velocity, okay? And this is all 14 sites. I wanted to look at just the west branch because that's seemingly the clearest and the most severe loss. And when you do that, just the west branch, we're up to 72%, almost 73% of the variation in biomass is explained by water velocity in the west branch. Incredible, really. Surprising to me. Okay, so this is just that I chose the marsh edge because those are the ones that are really receding. You can look at other areas up the marsh and you sort of see a similar trend, but the marsh edge is really important. Okay, so clearly hydrology matters, both inundation time, but water velocity probably most important, which if you go back now and start looking at the history of salt marsh ecology, it's one of the first things that Redfield talked about in his 1940 paper about the evolution of a salt marsh and how it grows, is that the hydrology and water velocity matter. You're not expected to see salt marshes in fast-moving water. They live in quiet bays and estuaries for a reason. Okay? So, it's probably something to do with sediment dynamics, and we want to do some more looking at sediment dynamics. What's the rate of depositions, how that changes, and how erosion is actually occurring. But now what next, right? Now we have some data, we have some really good understanding, I think, of these marshes. So maybe it's time to step in. Maybe now is the time that we know these marshes a little bit, we've done some science, we can step in and do a restoration maybe, but slowly. So my recommendation, as you'll see in a second, is we need to now experimentally do this, and there's some reasons for this. There's a lot of talk of thin layer, which is dumping dredge spoil or some amendment onto a marsh to keep it up, keep that marsh accreting up with sea level rise. Okay? So you'd spray the sediments over an area, usually it's dredge spoil, but not always. Okay? And that basically gets mixed with water, you spray it all up there, you've raised the marsh elevation a little bit, and then maybe your plants come up through that, or maybe you have to go back and plant. Okay? And then that just buys you some time with sea level rise, essentially. You've brought the marshes up a little bit faster than normal. What you're doing is essentially mimicking the natural cycle of a marsh, right? So this would be a healthy marsh, it's growing, accreting those sediments, and as it starts to go under and converts to a mud flat, right, if that was what was happening here, we'd step in, we'd spray that thin layer on top of it, right, and then now the marsh would colonize that bare area and kind of keep up, and you'd be that much ahead. You accelerated that natural process. Okay, perfect. Would work great. But not if you haven't done anything with the hydrology. If you've lost your marsh because water's ripping it away, you do this, water's just going to rip it away again. So that's a problem. How do you keep that sediment where you just put it, right? And then what about the marsh morphology? And I'll talk about that in a second. Okay, so this has been really successful. And if you go Google, look around, you can find tons of thin layer successes. There's lots of them. Jamaica Bay in New York was a huge, pretty good one. 2 acres, right? They went out and sprayed it, they planted it, they monitored it. It was kind of done experimentally. This is monitoring for the actual basic layer, the height of the marsh. They put these rods in, you go back and keep doing it. You can see whether the sediments are staying there, if you've lost them, or if the marsh is starting to accrete again, which would be the best case scenario. So that one's worked. Many of you may have heard of Ninigret Marsh down in Rhode Island. Massive, massive undertaking. I mean, you can just look at all the organizations involved, right? Lots of money, I think it's $2.2 million. They dug up and spread dredge spoil all over this area. You can sort of see it here in white. A couple things to note. There's that nice border along the edge. This is all high marsh restoration. They're not doing the Spartine alterniflora edge than what we would need to do here. They're spraying it up the uplands and raising that up because it was either not keeping up or sinking or filled for some other reason and coming in. So you can see this sort of edge isn't touched all the way around. Okay? So, but another fairly successful, I think 2017 this was done. I don't have the date on there. It's done in 2017 and they're now monitoring, and it seems like that mid to high marsh level is doing pretty well. Okay? So it can be done, different in different areas. So what's the downside? Why not just jump in and start spraying our marshes with dirt? Alright, well, the cost of getting it wrong is huge. Not only for any of you that wanted to fund this project, but you get it wrong, you've wasted time, money, resources. But more than that, you become one of the things that everyone points to of, oh, there's restoration, but it was useless. It is hugely damaging. So when you do restoration, you want to make sure you have your ducks in a row, get it right. Okay, so what does that mean? Well, we need to think about site selection. Where do you do this? What areas of the marsh are you going to do it? How big of an area might you want to do? How much sediment do you put on? Is an inch okay? 6 inches? Do you put a foot? That could be, who knows? Experiments are going to drive that, right? You do a small test and you say, okay, 1 inch did this or that, it enhanced growth or didn't, right? So the thicker, it might be great. You buy more time, but not if nothing grows there. Other adverse impacts. There's a new thin layer up in Boston that used dredge spoil that now they didn't test the dredge spoil before they sprayed it. Unfortunately, now they've got high levels of PFAS, thank you, all over the marsh. Right? So you just, and that's because it's everywhere, but when you spray it, you put it on the surface, it's now exposed to all those plants and animals that you've just accelerated its transition to the food chain. Heavy metals, which are commonly trapped by salt marshes and in submarsh sediments, right? You're spraying it on top. So these are all things you need to maybe think about before going this route, right? Why not? Let's do it right. And there's also some changes in soil chemistry by adding that layer. You get sulfides, which are really bad for the root system and also affect nutrient uptake for salt marsh plants. So there's that. Okay. Once you spray, you might have to plant. Another cost we'll talk about in a second. So our marshes here are a little different. Hydrology matters, I said. So spraying it and it gets taken away is not a good idea. So what can we do to maybe slow things down or at least shore up that shoreline? And there's this other type of restoration that's called living shorelines. Been used mostly in the mid-Atlantic but in other areas to preserve coastal habitats. So one of the ideas is work with biology that's there. So oyster reefs are really common, mid-Atlantic. They basically use oyster shell, bag it, and deposit them along the edge. And then that gets consolidated by the roots and sediments that it traps. And then you basically buffer that shoreline. Okay? So what we would have to do here, and there's different flavors of this, is called the marsh tow revetment, which because of that steep wall we have here and that undercut, we would need to support it, bag, clam shells, oyster shells, Gurkensia shells, whatever you can, or rock, and stabilize this edge from collapse, right? And then maybe, once you've done that, you could do some kind of thin layer on top of it if that's needed. But more important here is that collapse. But even this isn't going to affect our hydrology. Water's still going to move by it. So, but it might protect this edge, and then we might not get anything in that along that sort of oyster shell area, but it might at least protect what's in behind it. Okay. When you consider doing a thin or a shoreline project, I sort of listed what everyone thinks are the most important factors, and I went through it and I was like, wow, we already have data on some of this stuff. This makes us a huge step ahead. We know tidal inundation at all our sites. We know current speed. We have an estimate kind of wave energy, and we can get more refined details on that. We know the water depth. We know the proximity. The extent of erosion, we have data on that already. The cost we can possibly work out. So we have some of the data already from the work we've done. So if we wanted to go about this, we're that much further in planning, which I think is great. Okay. Just to give you guys an idea, typical cost of a living shoreline revetment, we're using oyster shell, which I don't know if we can get here as easy as they get down in the mid-Atlantic, but you know, you're looking somewhere about, there it is, about $3,500. That seems really cheap to me, but this is out of the literature, for 50 feet of shoreline. Okay, so oftentimes this is sold to homeowners down in Virginia. If you want to protect your property, here's what it's going to cost you to do. If you want to use rock, it gets a little bit more, and then the plantings on top of that go up depending on what species and who's growing, and can you find like a marsh donor, right, basically. There's lots of permitting questions. I didn't even get into that side of it. There are a slew of permitting questions within this restoration. All right, so where are we now? We have these marshes, we have the background. I think we now have the science to start investigating on a controlled experimental way restoration. I think we put ourselves in that thing. We're not going to be Ninnegrit Pond where we're like, right? But I think we can do a few more things and figure out what would work, and then figure out what do we want to do. I think those are two really important questions. So it's, I think, with science and understanding, we have the ability now to conserve and protect our marshes. Okay? I don't know how long for, but I think we can do something rather than now sort of, you know, some people say sit back and watch them just wash away. No, I think now we sort of have an understanding of what's going on, that we can do that. So I am going to just put this up and thank, one thing I forgot to thank up top is property owners. Thank you. If you're here, you make this work possible.
Yes.
Without property owners letting me walk across your marsh, or drag my wagon across your marsh, or you're parking your driveway and all those kind of things, we literally could not do this. Because sometimes I can't go by boat because of tides, and sometimes I can't go by boat because of time, which is more important to me sometimes because I'm teaching a class. I run out here, I can drive down your driveway, park at your house, run out in the marsh, get back to Providence and teach a class. That's invaluable. So if you're one of the property owners, thank you, thank you, thank you. And I'm gonna hand it to Mike because he's got a few things to say about the additional value of doing the tide gauges, and I think a couple other things, right? So then I'll take questions if there's questions, but I want you to listen to Mike for, I know, sorry.
For a minute.
First of all, Patrick, that's just Patrick is a much better scientist than he is a businessman, because he doesn't charge us anything for the second phase of the study.
Never.
I have outside here, before I get to it, lean up against the frame of the canopy, you can see one of the tie gauges. And attached to it, you can see the 4-foot ruler there, attached to that is one of the flow gauges. And just so you know how it works, if you're interested, that thing that's dangling off of the rod sticking out, that just goes with the current, and that device measures the angle of things at, it knows how fast the current's moving, and it also knows, because of its magnetics, it knows the direction. And the depth is measured by a pressure sensor right around where that white piece of tape is, which is where, that would be where the river bottom is. And the computer's up top, in the orange part, some of you can't see that, that's where the data gets collected. So, I didn't do all this by myself, I have a lot of help. I'm just gonna call out here, if you're here, I want you to raise your hand, okay? This is Sean Leach, he actually helped out with this kind of special geolocating work that we had to do to tie all the gauges together. Oh, all right, let me back up. Patrick mentioned that I had gauges in places that were not necessarily where he would want to put them. So we had 4 gauges, 2 on each side, a quarter mile and a half mile north and south of Hicks Bridge. Okay, and what we wanted to do there was we wanted to try to see, we're trying to understand all the hydrodynamics in the river, but we specifically wanted to see if this bridge, or the remnants of that bridge, are really affecting the tidal flow and the flushing of the upper east branch here. So, this is a little summary, it's a little technical, but if you start at the mouth of the river, and this is right here, at Boathouse Row, and then you proceed up the river along this blue line, past trips, under the bridge, under Hicks Bridge, all the way up to the head, you're traveling along this line right here. And you can see at the mouth of the river, we have, let's say it's 3 feet of tide. So we have 100% of the tide at the mouth. But as you start to go past the mouth, the tide drops, say from 3 feet to 36 inches down to 32 inches down here. That's an average. And then as you start to go up the river, we have a little, like a small resonance, like a little Bay of Fundy effect. It doesn't really bring it back to 100%, but it starts to grow as you go all the way up to the head. What's interesting here is this red line is Hicks Bridge. Something's going on there. We're just uncovering this now. We're going to employ, get some consultants to help us figure out, you know, is this a result of the bridge and the rubble? And would it make sense then to take the rubble out? And would there be a benefit that's worth the money that it costs? So, let me jump into all the folks that have helped out. You saw Mark already, he got his award, this Patrick. Okay, here's Pam Beck, she's not here today, she helped out on a really crummy day out there on the marsh. There's Yope, and this is Bobby Carroll, he helped me out, he was in the water actually, he helped me out 2 years ago. Here we have Paul Mooney and Harold Cooper. He's not here today, he's in Arizona, but these guys helped out on several sites. And we have Ken Short. You're supposed to raise your hand. Where's Ken? Come on, raise your hand. Okay. Sam's not here today, he helped me out on a site. And here we have Jim and Wendy. How about those hands right there? They helped me out 2 days doing the survey work. And then we also, we did another little project we didn't talk about, but we've got sensors on either side of the causeway out in the underwater on the bottom. You can't see, no buoys or anything. But Mark and Jason Powell helped me go out and deploy those sensors. And unfortunately, it's still out there over the winter, so we got to go out in the spring and pick them up. Kerry Fennelly's the guy that owns Westport Sea Farms. When our boats are out of the water, he has to help me out. So he brought me over on the other side of the bridge there. So anyway, lots of help. This is Sean again, and the other fellow, Matt Farias, that helped us with the special geolocating. 3 people I didn't have pictures of that helped out, but with the assistance of AI, I just put the question out there. And what do you know? Kim, Tom, and Paul, 2 of them here, raise your hand, yes, Barry is there. Well, you know what? We found the pictures, okay? They really helped.